Sorry about that.
I guess we are on now. Is that true, Anne? Can we start?
Yes, you are on and you can start. Thank you.
Okay. Fantastic. Welcome everyone to the plenary session number 3 on clinical trials of this year's virtual meeting of the EHDN. I think it is an exciting time with a lot of clinical trials and other studies that have read out that are ongoing, and we will hear updates today from prestigious representatives of each of these projects. My name is Dr. Ralph Reimann. I'm the chair of the George Huntington Institute in Münster in Germany. And I'm joined here to chair the session by Dina, who will now introduce herself and introduce the first speaker, and we will switch back and forth then. So go ahead, Dina, please. And by the way, questions can be asked in the panel discussion, which will follow this section. So we'll keep it at 15 minutes for every speaker. Have the speakers speak now, and you can already enter questions in the Q&A, but we will answer them in the panel discussion. So Dina, please go ahead.
Thank you, Ralph. Hello, everybody. My name is Dina De Souza. I am a board member of the European Huntington's Association, and it's a privilege to be doing this for the second time with Ralph and co-chairing the clinical trials. So we'll start off. We're going to have Dr. Scott Schobel first, and he is Group Medical Director and Clinical Lead of the Roche Tominersen Global Development Program, and which has been for Scott the privilege of his lifetime. Previous to that, he was an academic in New York studying hippocampal dysfunction in neuropsychiatric diseases. And Scott enjoys keeping up with his 3 sons, learning German, and all forms of music and dancing in his spare time. So I'll hand it over to you, Scott. Thank you.
Thank you, Dina. It's a great pleasure to be here. Let me just share my screen quickly. And you can hear me, I trust. And I will share my screen. Can you see that?
Yes, very well, Scott.
Coming through? Okay, great. Good day, good afternoon, good evening, wherever you may be in the world. On behalf of my colleagues from Roche and Genentech, I'm pleased to provide an update from the Tominersen Clinical Development Program. It's obvious, but just a mandatory disclosure, I am a full-time employee of F. Hoffman La Roche. And I want to always acknowledge our steadfast collaborators in the field, including first and foremost our partners from Ionis, pioneers of ASO technology, and creators of Tominersen. Many of the institutions here who we've enjoyed deep and ongoing collaborations, the EHDN for obviously everything it does on this side of the pond, as it were, as well as this meeting we're all at virtually today. I just want to also call out, there's one individual pictured here in the lower right-hand side of this slide, and that's Peter McColgan. And we're absolutely thrilled that Peter has been able to join us on the Tominersen team and at Roche more generally over this past spring into summer. And you know, Peter's a neurologist by training, Huntington's disease specialist, who's a fantastic physician scientist in his own right. And he's really come on strong as we've been beginning and continuing this large scale data analytic effort, particularly on the brain imaging side, as well as the biomarker side, relating that, you know, sort of to all of the data we have on the outcome side. And I think it's just been great to have Peter come on board. And last but not least, you know, a deepest gratitude to the investigator network during this time on the program, the study participants, as well as their families. We've had unbelievable rates of adherence to protocols despite stoppage of study drug and pausing of study drug. And I'll say more about that in a minute, but just a real heartfelt thanks for that ongoing collaboration. What I'd like to do first is look back over the earlier part of this year and review that, and then kind of bring the group here to the current status of where we are, and then look ahead a little bit. So in terms of where we've recently been, looking back to the spring of this year, The Generation HD1 phase 3 pivotal study received a recommendation from the IDMC to stop study drug dosing in the Generation HD1 study. And we then quickly pulled down the data through the IDCC. We unblinded ourselves as a sponsor while maintaining the study blind at the site and participant level to protect the external blinding and integrity of the study, but chose to accept the recommendation to stop the tominersen dosing. And we presented those top-line results at CHDI in April. And we're incredibly thankful, and essentially what's happening now is the vast majority of participants, about 85%, are still in study protocols and are being followed for clinical and safety outcomes. This will allow us really to fully interpret the Generation HD1 study, which is great news in this recovery phase of the off-drug phase, and will help advance our understanding of Huntington lowering approaches, for not only tominersen, but should be useful for the field as well. And that clinical and safety follow-up is ongoing as we continue to look at this data we've looked at in the spring. So obviously, this effort is in the context of going forward in time now to today in early September. And but let me just quickly review the spring in terms of those findings, and then we'll go to the current status. So Gen HD1 did show a consistent pattern of clinical progression over time in the key outcome measures, so we could interpret results that enabled the IDMC recommendations to occur. And essentially, the long and the short of it is that the 120 milligram Q8 weekly regimen showed an unfavorable benefit risk profile based upon unfavorable efficacy versus placebo. And the 120 Q16 week, though having a safety profile comparable to placebo, showed no apparent observable benefit at a relatively late time point in the trial at the 17-month time point. So overall, looking at this holistically, the IDMC made the recommendation to stop study drug dosing. And we took that recommendation, and so we stopped dosing in Gen HD1 and paused in Gen Extend based upon those recommendations. So that's the spring. So where are we now? So first and foremost, the IDMC decision came as a surprise to us, and what it resulted in was basically a tsunami of samples available suddenly for analysis when we decided to unblind ourselves and look at the data. Over 40,000 samples were suddenly available for analysis. So we made a plan to ship samples to our analytical centers. We had always planned batch analyses for certain fluid biomarkers because these are largely immunoassays, which are notoriously finicky when it comes to running them from batch to batch. So we wanted to get them all within the same batch runs to the extent possible. So we made an elaborate plan to ship samples of our key biomarkers of interest, including CSF mutant Huntington, CSF NFL. We've been sending PK samples all along, but this was a large effort. We then have been starting to clean and lock databases based upon this information as it comes in. We've then, of course, begun to have the vendors analyze and send to us those outputs. And we have just begun, literally, over the last several weeks, what is sort of a more simple-minded single outcome factor analysis. So you look at the time course of one biomarker versus another, in the active arms versus the placebo arms. And we've been sort of at this more descriptive level in our recent analyses. And we're looking forward to now going to the next level, which is a more integrative approach, where we're looking at the relationship of change in one biomarker to another biomarker outcome, or a change in a biomarker outcome to a clinical outcome. And this is the more integrative view, which is frankly not complete. A lot of that hasn't been done yet. So what am I talking about when I talk about these analyses and what we're looking at? So what we're looking at are clinical outcomes. We're looking at them at baseline, parameters such as disease stage status. Frequently, a CAP is the CAG-Age project is used in this regard. We're looking at exposure, and drug concentration. We're looking at the fluid biomarkers I mentioned. We're looking at MRI, as well as the digital outcomes we've put in most all of our studies, including GenHD1. And we're also exploring genomics. And our key point is integrating the understanding of what are the relationships between the different data and variables. What does this mean for tominersen? And what does this mean by extension for Huntingtin lowering approaches? So we are just really getting into the thick of this as we speak, and essentially brings us back to sort of today on the timeline. Now, after we do all of this, we have an internal and external step to consult with experts and regulators. We've begun to consult with our steering committee on this information as it becomes available. We're also looking forward to discussing it with regulators. Thereafter, I believe that we will have, and I'm confident that we will have, to the best of our ability, an integrated understanding of this data, which can then be shared with the community. And this analysis plan has been all predefined. We spent several weeks post CHDI top line sharing of results, generating this analysis plan. It has several thousand outputs, many samples. A lot of people are paying full-time attention on this task. And as soon as we have our integrated understanding, we have had it externally and internally vetted. And as soon as we know where we're going with on a program level for tominersen, we will be planning to share this with the HD community. In terms of the timelines of that, I am looking, and I've discussed this with the group internally. I'm looking at the first available, safety information from the follow-up of the participants towards the end of this year, and the rest of it probably into early next would be my best guess. But I can't get more specific than that because we simply won't know when we're fully integrated and aligned until we know. And as soon as we do, we'll be sharing this with the community. And I just want to say also, I think about this most every day from the perspective of an individual living with HD or a family member, just how hard it is to wait. And I want to give you a very clear message. This is not because we are holding information or there's any good or bad information. It's all kinds of information. And the issue is really, we don't want to put out partial information because we think that could lead to then expectancies that we're not sure we can deliver because we don't have an aligned company position yet on where we're going next. And so it's really the idea to get it all looked at, get that position, and then come out all at once. And we think that's, in our view, doing a service, to the community. And it will be very good when it's ready, and it should be a very good set of new understandings for the field. In terms of what we're looking at, again, just to hit the real core highlights of what I think we can deliver when we are ready. On disease stage, we are looking at the disease stage baseline status in terms of mediating treatment response to tominersen. We suspect this is going to play a role. We're looking at the drug exposure levels. No one had taken in a dose of 120 milligrams of an ASO full stop in a chronic experimental paradigm. And we do think that that may have been too high as our best sense, at least judging from our early program experience, where we once have already backed off frequency of the drug administration. But there is this overarching issue of what about the drug exposure? Are there optimal levels of exposure among the participants that would lead to a more favorable benefit risk profile? We are looking at this with a fine-tooth comb and believe there'll be something coming out of that. Patient characteristics, are there individual patient characteristics which predict response to tominersen? And finally, on the mechanistic understanding front, how do biomarker patterns inform our understanding? What is the role of wild type lowering or determinants of ASO tolerability? Probably the hardest here is the wild type lowering because we don't yet have an assay that we've been able to optimize that is ready for high throughput sampling on the order of magnitude that GenHD1 requires. So this will remain sort of more of the role, in our view, of preclinical or translational experiments. Perhaps that's a future avenue we could look into. But at least in terms of these first three categories, disease stage, drug exposure, and patient characteristics, I believe that we will have good answers to these questions when they're available, which can meaningfully guide not only tominersen, but the community. And so in this sense, no research effort of any participant on tominersen, despite the uncertain status where we are today, is unwasted. It's my best sense that we're going to be able to deliver answers on that, which will be meaningful and impactful for the field. And I just want to finish to say a big thank you to the community. Again, like Dina said in the beginning, it really has been the privilege of a lifetime to continue to be a part of this program. I'm extremely happy to have been joined by Peter. Our team continues every day with hope and enthusiasm to see where this story is going to play out. And you know, the ongoing contributions are very inspirational and valued by us. So with that, I'm going to stop and thank you, and I'm happy to turn it back to the session moderators. Thank you.
Thank you, Scott. That was very useful information and concise. So answers still to come, and hopeful about the outcomes. So
thank you.
Ralph?